Hello students, myself Popere KS, Assistant Professor Botany of Raj Shikshan Sanstha's RBNB College, Srirampur. In this lecture, we are going to study about the different types of RNA which is present in living cell or the cell of the different organism. So first let us start with RNA. What is an RNA? So, what is RNA? RNA, it is an molecule that it is a single standard molecule. Like DNA, it is a double standard molecule. So, this RNA, it is a single standard molecule. Now next, it has a presence of different sugar from that of the DNA. In DNA, the sugar is deoxyribose, but in the case of RNA, the sugar is ribose. So in RNA, there is presence of ribose sugar. Now next is, it has presence of base which is different from that of the DNA. In DNA, there is presence of thymine, but in case of RNA, instead of thymine, there is another base which is called as uracil. So, there is presence of a different base, that is uracil, but in the case of the DNA, instead of uracil, there is presence of another base, that is the thymine. So in case of RNA, the sugar or the nitrogenous base is uracil. So these are the three properties of the RNA which is different from that of the DNA. So RNA, how it is different from DNA? That it has a single standard, it is a single standard molecule, it has presence of ribose sugar, and third one is it has uracil base instead of the thymine. Now next we will see the different three types of the mRNA which is present in a cell. So there are the three types of mRNA which is present in the cell are in a cell. First is mRNA, and the third one is tRNA. So these are the three types of the RNA which is present in a cell, that is mRNA, rRNA and tRNA. Now we are going to see the structure as well as the function of this three RNA, that is mRNA, rRNA and tRNA. So let us first start with the first mRNA, that is the mRNA. First is now this is mRNA. Now this mRNA, it is present in least abundant in the cell. So of the three RNA, this mRNA, it is present in least amount or the least abundant in the cell, that it will be from 5 to 10% in a cell. Now next, the structure of this mRNA, So this is linear in structure and it has polarity that is 5' to 3'. Now first there is presence of a codon. So there will be, it consists of this mRNA, it consists of different type of codon on mRNA. So mRNA, there are presence of different codon. Now what is codon? Codon is it is made up of 3 nucleotide. That 3 nucleotide, it makes a codon. So this codon, they are present throughout the mRNA. Now it has presence of codon, that is start codon and stop codon, or initiation codon and termination codon. So AUG, and stop codon will be so these are the start codon. This is called as start or initiation codon, and this UAG, UGA, or it can be UAA. These are called as stop or termination codon. So this mRNA will consist of a start codon and a stop codon, and in between this codon there will be presence of a region which could be called as a coding region. So in between the start and the stop codon there is presence of coding region, and in this region will be coding for the synthesis of the protein. Now this mRNA, at this start site there will be, this is the site from which the protein synthesis process will start, and at the stop codon the protein synthesis terminate. Now this mRNA, it is acting as a messenger of DNA. So it will carry the genetic information from that is for the synthesis of the protein. And this mRNA, they are important for the transfer of information. So this will be involved in protein synthesis process. It will act as a template for protein synthesis. Now this mRNA, they can acquire secondary structures. That the secondary structure which will be acquired by mRNA will be the stem loop or the hairpin loop. So they will acquire secondary structures which could be a hairpin loop or a stem loop. So this is a secondary structure which will be acquired by this mRNA. This will be called as this is called as stem, and this will be called as loop. So they will acquire a secondary structures, that is stem loop or hairpin loop. So this is about the structure and the function of the mRNA. Next we will see the structure and the function of rRNA. So next is the rRNA. this mRNA is messenger RNA, this rRNA is ribosomal RNA. Now it constitute means it will be present in a cell in a bulk amount, or it constitute the bulk of the cellular RNA, that is it will be present in bulk, that is up to 80%. The total mRNA which will be present in the cell, it constitute of the 80% of the total RNA which is present in a cell. Now it is large in size and they are of various type of rRNA which is present in a cell. Now we will see means they form the most complex structure mostly they will be formed in a folds and loop. So suppose this is how this mRNA, they will form the secondary or the tertiary structure or the most complex structure. They are formed by the rRNA. Now this rRNA molecule, they are mostly present in the ribosome. So these are mostly present in the different subunit of the ribosome. Now in case of this ribosome, they are different in eukaryotes and prokaryotes. So in eukaryotes, the ribosome, it is made up of two subunits, that is the and 30 subunit. This larger one is of 50 subunit and smaller is of 30 subunit. And in prokaryote, this is in the case of the prokaryote, and in case of the eukaryote, it will made up of two subunit, that is the 60S plus 40S subunit. So in this subunit, there are presence of different rRNA. Suppose in the subunit, that is 30S subunit of prokaryote, there is presence of 16S rRNA. So it will have presence of 16S rRNA. So there are different types of rRNA which will be present in the ribosome. So it will be 16S rRNA, 23S rRNA, 5S rRNA. So there are various types of rRNA which is present in the ribosome or in the cell of an organism. Now in case of the prokaryote, there will be 20S rRNA or 18S rRNA, then 5, that is 5S rRNA. So these are the different types of the rRNA which is present in the cell. Now this rRNA, it is also called as a ribozyme, which will do the process of protein synthesis, or it will carries this ribozyme or this rRNA, this will do the synthesis of the protein, or it will function in the protein synthesis. Now one more term, this 16S rRNA which is present in the 30S subunit of the prokaryote, it is one technique is there, that is 16S rRNA sequencing is done. By this technique we can, or the bacterial classification scheme was developed using this 16 rRNA sequencing. So this is about the rRNA. Next we will move to the last type of the RNA, that is the tRNA. So last one, that is tRNA, or it is called as transfer RNA. Now this tRNA, it is the smallest of all. And it is means there are different types of tRNA which is present in a cell. Now this tRNA, one tRNA will code for the one type of the protein, or the one type of tRNA, it binds specifically to each of one amino acid that occur in a protein. Now this tRNA, it is a single standard and it will consist of 73 to 93 nucleotides. So it is made up of 73 to 93 nucleotide, and it will undergo folding, or it will form the tertiary or the secondary structure because of the internal complementary base pairing, and it will acquire a L-shaped structure or a tertiary or the secondary structure. Now we will see the structure of the tRNA. So this is the structure of the tRNA. It consists of the various loop and the stem. So this is D loop, this is T loop, this is acceptor arm, this is variable loop, and this is anticodon loop. Now this as acceptor arm, it will consist of 3' and 5' polarity. Now this 3' end, there is a hydroxyl group, that is OH is present, and at the 5' there is a monophosphate present, that is P will be present, that is a monophosphate is present at the 5' end. Now this acceptor arm, it has presence of 4 base pairs and 7 nucleotides. So this acceptor arm, it has presence of 4 base pair and 7 nucleotide in all, and it also consists of conserved sequence, that is CCA. So it also has a conserved sequence at the 3' end, that is CCA region, which is conserved. Now next is the TC loop. This loop, it is consist of the 7 nucleotide in the loop region and 4 nucleotide at the stem region. It will consist of the 5 base pair. 5 base pair in the stem region and 7 nucleotide in the loop region. Now next, this anticodon. At the anticodon loop, there will be presence of a codon, that a codon means a 3 nucleotide will be present. So this anticodon, there is presence of the different codon. Now this codon, when this tRNA, it will charge or it will carry a amino acid, or it will carry an amino acid for the protein synthesis. There will be a reaction of this anticodon and the codon, which will be present on the mRNA. So there will be a presence of codon. Now it has 5 base pair in the stem region. This and the stem of the anticodon, it has presence of 5 base pair. This variable loop, it is variable in different tRNA, and it may be from 4 to 5 nucleotide. 4 to 5 nucleotide may be present, and it is variable, so it might contain, that is 10 to 24 nucleotide, that is in the different tRNA. Now this D loop, it is variable in different tRNAs. So this loop is variable in different RNA. So this is about the structure of the tRNA. So this tRNA, it is acting as it is carrier of the amino acid. During the protein synthesis process, there will be reaction between this anticodon and the codon, which are present on the mRNA. So suppose here is presence of UUC. Now this will pairs with this is an mRNA. Now this will have C and C. So there will be anticodon codon reaction, and it will result in the synthesis of the protein. And this tRNA, this is important for the protein synthesis to occur. So this is about the 3 types of the RNA which is present in the living cell, that is the mRNA, rRNA and the tRNA. Thank you.